Yeah, so I'm going to kind of building on what Israel and Brent have introduced, talk in a little more detail about a case study focused on a conservation prioritization for the plants of California that uses these ideas of phylogenetic diversity and these comparisons among diversity facets to try to identify the most important future locations for new reserves across California. California is a super important place to be thinking about creating new protected areas. First of all, it's a hugely biodiverse region. It's considered a world biodiversity hotspot. This map here is showing the richness of US state endemic plant species. Obviously, California pops out here. It also pops out for a lot of other taxa and in a global context. So it's a place where there's a lot of biodiversity we really want to be protecting. Next slide. It's also a place where we have an unusually detailed and rich amount of information about the biodiversity. So for plants, for example, there's about 5,000 species of plants in California. We have really good data on the geographic distributions of those species across the state, as well as on their evolutionary relationships. So that means California is both sort of a good test case for developing and testing ideas about representing biodiversity and comparing different biodiversity metrics. It's also a place where we can put more credence in the results of our spatial biodiversity analyses in terms of using those results to actually inform conservation on the ground. Next slide, thanks Israel. And then it's also a place where there's both a really large and growing human footprint. So this map on the left shows landscape intactness, or so how ecologically pristine different parts of California are. California has a huge population, a lot of urban encroachment into wildlands, and that poses a real and ongoing threat to its native biodiversity. It's also a place where there's historically been a lot of conservation activity and a lot of protected areas created in the past. So in the middle map here, we're looking at a map of the degree of protection of different areas across California, with the yellow locations being the most protected and blue being the least protected. Land protection is not a binary variable, right? It's really a continuous gradient. We need to be thinking about that continuous gradient of protection status as we work with conservation prioritization, right? Because if we upgrade, let's say, a private, totally unprotected piece of land to full protection status, that's going to be a much larger protection boost than if we upgrade, a piece of national forest land that's already partially protected in terms of the management activities and the biodiversity goals there. So when we combine these two maps, what we see is that a relatively small fraction of California is left that's currently unprotected, that has high ecological intactness. And so it's a really high priority to think about creating new protected areas in these remaining areas of unprotected intactness and figuring out what the highest priority areas are to focus the creation of new reserves before they get further converted to development or agriculture. So our key questions for this analysis were, first of all, where should we be placing these priority areas to protect California plants in the future? And then second of all, how do those priorities differ depending on how we quantify biodiversity? How does species richness priorities compare to phylogenetic diversity, and how do those three different facets of phylogenetic diversity that Israel discussed compare in terms of the different locations they suggest as being most important for future reserves in California? Next slide. So here we're looking at yet another view of these three different facets of phylodiversity. We can see that, the taxa, the species that are at the tips of these trees, and all the internal branches. If we consider the branch length sort of a metric of their conservation value of how much unique biodiversity each one of these taxa contains, we see very different patterns in terms of which one of these branch lengths we use. And as Israel discussed, these three branch length metrics are associated with different conservation values and different goals in terms of, do we want to conserve species that are most likely to be resilient to future environmental change? Do we want to conserve species that are the most diverse in terms of their genetics and their physical traits? Do we want to conserve species that are most likely to be able to regenerate biodiversity in the future after a mass extinction event? So we have these three different ways of looking at biodiversity. Israel, next slide. And what we did here is a big statewide data analysis focused on the full flora of California to try and develop this set of conservation prioritizations. So we created a phylogeny and an evolutionary tree of the plants of California. We created species distribution models for all 5,000 species of California plants. And then using the phylogeny, we also merged those models to create distributions for all of the larger level clades. So we have 10,000 different taxa, different levels of the tree that we have distribution models for. And then we also developed this new data set showing the current protection status of every different location across California. Next slide. Now, if we just look at the raw biodiversity patterns, sort of without considering conservation explicitly, there is a lot we can learn just placing every community of taxa across the state somewhere in this three-dimensional biodiversity space. This is all very interesting from a sort of basic biodiversity and biogeography standpoint. It's a little more academic. If we want to then add a layer of conservation explicit prioritization on top of that, we have to also consider some additional attributes of these taxa and landscapes in addition to simply their branch lengths. So next slide. As we're trying to identify priority sites, there's a number of things we want to consider. So first of all, we're going to use our range models to consider the probability that at any prospective conservation reserve site, each taxon is actually present, right? This is a probability, not just a binary presence or absence. We want to consider the branch length of each of these taxa for all the reasons that Brent and Israel introduced. We also want to consider how important that prospective reserve site is to each taxon, and that's where range size or endemism comes in, right? Because if you have a really small ranged taxon, then each location, every population where that species or clade occurs is going to be really important to that species. And by protecting that site, you're going to be able to get a huge conservation boost for that species. Whereas a wide-ranging taxon, a small reserve on part of its distribution is not going to make a huge difference for that taxon. So that's where the endemism or range size piece comes in. We also, of course, want to think about how much of each taxon's range is already protected and emphasize creating protected areas that capture the distributions of taxa at every level of the phylogeny that are currently poorly protected. Again, we want to consider the level of protection of each site across the state as a continuous metric. And then we want to think about the complementarity of sites to the current reserve network and to other priority sites that we've identified, right? So it's not just about thinking about how much biodiversity is in each individual location and trying to find the locations that have individually high levels of biodiversity. It's trying to identify a set of locations that collectively capture as much of the evolutionary tree as possible. And that turns out to be quite an intensive, computationally intensive problem, because you have to evaluate the biodiversity value from millions of potential reserve networks. Next slide. So there's different approaches for this. We use an algorithm called stepwise optimization. And the way that we set this up, we're identifying sort of an ordered ranking, a set of priorities for locations to create new reserves. And these new proposed reserve sites are places that are currently poorly protected, that contain lots of taxa at every level of the tree, that have long evolutionary branches, that have small geographic ranges, and that have poor protection currently across their ranges based on existing protected area. And then the set of proposed reserves collectively complements both each other as well as the existing set of protected areas in California. And when we run these, next slide, that's fine. This is what we get. So this slide is showing a comparison between the top 50 most important priority locations for new reserves for species in pink and red, and for phylogenetic diversity for when we consider evolution, which is in like light blue and green. We can ignore the OTU thing for the purposes of today's talk. And what we see is that there are major differences in terms of what part of California get identified as the key regions, as well as specific sites for creating new reserves, depending on whether we just look at species or whether we consider the relationships among those species and how unique they are. Next slide. We can then also, in a similar way, look at the top 50 priorities for the 3 different facets of phylogenetic diversity. And again, we see a large number of locations that are in all the various colors, where only 1 or 2 of the 3 facets of phylodiversity have flagged these sites as key conservation priorities. So that means that it is going to make a difference which one of these aspects of biodiversity we choose when we're creating priorities, and it is going to make a difference at various spatial scales as well. The good news, though, is that there are a lot of sites which are in black in this map that are identified as top conservation priorities under all 3 of the phylodiversity facets. So that's good news, because that means there are a lot of places where no matter which one of these branch links you have that's associated with different sort of evolutionary attributes and different conservation goals, those are places where we can agree they're definitely high priority sites. Next slide. And those include a number of well-known plant biodiversity hotspots across the state, including parts of the Sierra foothills, some spots along the coast, both in Northern California near Humboldt Bay, as well as in the Point Conception region near Santa Barbara, areas of the Inner Coast ranges in the Mount Hamilton range, and other spots. And the great and exciting news is that in the time since we've published this paper a few years ago, 2 of these have actually become protected. In Mount Hamilton range and the Point Conception area have been added to the protected area network through collaborations between public and private and nonprofit groups. So really exciting to see all of the progress and the energy that's going into creating new protected areas in California, and to see the passion that the conservation community has for trying to identify these conservation or these biodiversity hotspots and protect them before we lose this biodiversity. So just a couple quick summary points here. We can definitely make major gains for protecting biodiversity by just conserving a small number of key sites that complement existing reserves across the state. But it does matter whether we quantify biodiversity as just species richness or whether we consider how unique and related these species are. And it also matters which one of these evolutionary metrics we choose. And then just more generally, thinking about phylogenetic diversity as a way to capture not just the existing patterns in the landscape, but also the process that generates that biodiversity, and to account for that in our conservation prioritizations as well. Next. So this is just a bit more info. A lot of these data sets that we developed are part of an interactive web app that's up online at that URL. This is published in an academic paper that you can check out as well. And so, yeah, if you want to switch to the discussion slide there, and we can open it up for any questions folks might have. So I think we have touched on a lot of these points already. The phylogenetic piece essentially allows us to make sure that in our reserve networks, we're not just protecting a bunch of species that are super similar from one another, right? When we think of biodiversity, what we want to do is capture a diverse set of organisms. And that means not just a large number of species, but species that are different from one another. We don't want to just protect 1,000 species of fruit fly. We want to protect 1,000 different species that are as different from each other as possible, that we all sort of intuitively think of as what biodiversity actually represents. So that's why the phylogenetic aspect of biodiversity really makes a difference for conservation. The reason they give different results in some places and similar in others is simply because of the sort of uneven way in which species with different branch lengths and clades with different amounts of evolutionary richness are distributed in space. If everything was completely random, we would expect species richness to correlate perfectly with phylogenetic diversity, and the evolutionary piece wouldn't make a difference. But because taxa with long evolutionary branches are concentrated in some parts of the state, and taxa with short branches are concentrated in other parts of the state, and rapidly diversifying or slowly diversifying taxa are similarly distributed in different locations, we can get these very different results depending on which biodiversity metric we use. And then, yeah, certainly curious if folks have thoughts about additional factors that one would want to take into account in conservation prioritization beyond what we've discussed here.
Gary has a question in chat. Matt, if you can see chat.
Yeah, I see that. That's a great question. So the best solution for this is certainly to simply include these other taxa in the evolutionary tree as well, right? I mean, if you go deep enough in the phylogeny, obviously we can put all of these species, plants, birds, insects, or mammals, on the same evolutionary tree and simply do one analysis that takes into account all of these species together. Another option is to do separate prioritizations for each of these different taxa. Often there are concordances between these things because they're attributes of the landscape or of the ecological communities that sort of cause these things to be highly correlated with one another. But this is a really underdeveloped and underexplored aspect of spatial phylogenetics in general, is trying to compare and contrast and understand the relationships between phylodiversity patterns for different taxa. So I think this is really pointing to a need for more research and discussion for how we integrate these across taxa.
I agree with that, Gary. I would just add, and if you're interested in this, I urge you to do it, is one issue has just been convincing our colleagues that work on animals to do this. The botanist jumped out in the lead on this for some reason. And when I presented this to my vertebrate colleagues, they look at me and they go, huh, that's interesting. And then they just go ahead and do species. They don't get it or why they should do it. So if it's such a low hanging fruit, because the vertebrates particularly the data is really good and it's a really good phylogeny. I'm going to do it myself someday if they won't. But I just would encourage people interested in vertebrates to do it, because the insects have other problems, just that there are so many of them and they're not as good as spatial data. But there is one butterfly analysis out for the United States. We showed a quick slide of it at the very beginning. And I know of people interested in bees and in beetles that are working on it. So I think there may be some insect ones, but anybody listening, if you're interested in vertebrates, I just think vertebrates need to be done, and you could get a really nice paper out of it really quickly.
And I also think it's worth mentioning that there might, in terms of the mismatch between plants and other taxonomic groups, is because when we talk about coevolution and the way it is being explained, such that sometimes it starts with the plant evolving, and then there's a little bit of a lag period before there is a response evolutionary change for animals and other organisms that depend on those plants. So that also could also explain for some of the mismatch in the diversification or different patterns that we see between among different taxonomic groups.
Yeah, it would also be interesting to see where they align too, because just like where you see these different facets aligning in certain places, we could have areas where we have high PD for plants and insects and mammals and, whatever. And so then again, we can prioritize these as sort of high important conservation areas due to that as well, which is would be really neat.
All right, thanks, Matt. Thank you so much for that great presentation on applying spatial phylogenetics for conservation flora. And now we're going to switch it over a little bit into the next section, which will be on real case studies and applications, and where we'll be traveling all the way from California to Florida, and Julie will be helping us with that.